Hey guys, Mr. Dumber coming at you again from the Dunbar Corona compound here in the basement. We are going to tackle this last part of this chapter 12 and 13 PowerPoint. This time we're going to talk about mutations, which are changes in genes that could cause changes in the proteins that genes are supposed to make, which can really result in changes in traits and eventually even make you so different that you might have two species come about from one species. So mutations are mistakes made in the genetic code. Sometimes they're bad, sometimes they're good, sometimes they have no change, but they certainly lead to variety, and that's an important part of biology. Is variety is the spice of life. Variety is what allows us to all be different, and therefore, if we're all different, what kills one does not kill all. It also allows us to adapt and evolve over time. So don't think of the word mutation as a bad thing all the time, which a lot of people do think of that as. Mutations are just mistakes. They're just changes in our genetic code. And oftentimes, again, they can actually lead to good things, sometimes bad things, sometimes no real change. So let's start to tackle this PowerPoint, shall we? I'll get rid of my mugshot here. We hope you're doing well, by the way. We're almost done. I know it's been a crazy end of the school year, but we're almost done. Hang in there. Keep working hard. We miss you guys. So I'll be back at the end. All right, here we go. We talked about genetic code stuff already, and we talked about how your genetic code really leads to your traits, right? And we said that that's actually multiple steps to get from DNA all the way to your traits. But I think you guys remember that when DNA needs to pass its message off, it passes that message off to RNA, which is a helper molecule. We're seeing that right here. That process is called transcription. When DNA, the double-stranded helix here, passes its message on to RNA, and we said that's kind of like Miss Hager writing the pink slip. Well, today we're going to talk about wonder if there are mistakes in the DNA. You guys remember DNA needs to copy itself to make new cells. So sometimes when the copying process occurs, there are mistakes made. Maybe there's a G where there should be a T, or there's an A where there should be a C or something, right? And again, these are called mutations. Mistakes can also happen when we copy the DNA onto messenger RNA. That would be like Miss Hager making a mistake when writing the pink slip. And again, these changes are going to lead to a change in the protein and eventually could lead to a change in the traits, okay? So let's have a look at this stuff. We're going to scroll all the way down. I hope you're following along in your PowerPoint. We talked about this business of translation as well. So how do we take the genetic code and actually turn it into a protein? What do our cells do with this genetic information? And remember, it's all about building proteins from these subunits called amino acids. In last PowerPoint, we talked about how these different letter codes lead to these different amino acids. Very important point here. Sometimes you could have different letter codes, what we call codons, that lead to the same amino acid being made. So it's kind of like having two different spellings for the same word or the same name, such as John and Johnny and Jonathan. Really, they all mean the same kid. Or quick and fast. Really, those are different spellings, but they mean the same thing. So for instance, if my original DNA code led to this RNA code, let's say GCG, well, that GCG spelling would give me the amino acid alanine. And that would mean put alanine in a specific place in that protein. And again, that protein would be created and it would give you either a structural protein like muscle or hair or something like that, or maybe an immune cell, because those are also proteins or based on proteins. But it might also give you an enzyme which controls a chemical reaction that gives you a certain trait. But look at this. We could also have GCC. Let's say there was a mistake. Instead of GGG, we spelled instead of GCG, we spelled GCU. Well, guess what? That's also going to give us alanine. So that would be a mistake that didn't cause any real change in that amino acid to be in that protein. In other words, if Miss Hager put Johnny instead of Jonathan on the pink slip, she would still get the same kid. Okay. However, there are mistakes or mutations that are made in the genetic code that will lead to kind of large changes. For instance, if we wrote GUG Well, that's different. That's not going to give us alanine anymore. That's going to give us GUG valine. So some mistakes will lead to no change in the in the protein being made from the genetic code, and some mistakes will lead to small changes in the protein that's being made, and some mistakes will lead to very large changes in the protein that's being made. And all these mistakes can either be really insignificant, they can be kind of significant, or they can be huge. They can also be very good, really have no change, or be very bad. All right, so keep that in mind. Again, don't always think about mutation as being a terrible thing. Also, please keep in mind this translation wheel, this codon wheel, is just the way we have cracked the code. Human beings, scientists, have figured out what these genetic codes mean. They really just mean amino acids put in a certain order to give us a certain protein that will give us a certain trait. This chart down here is identical to the wheel that you just saw in the slide, 2 slides above. This is just a different way to represent the information. So I want to just demonstrate something for you here. If you looked at, again, let's go back to this let's do this, GGG. That would spell glycine. The way the translation wheel works, the codon wheel works, is you read from the inside, where the big letters are, towards the outside. And please remember, genetic code is always read in letters of 3, groups of 3. So 3 letters is equal to 1 genetic word. Very important. All right. So GGG gets us glycine. Keep in mind, though, again, this chart is the exact same information. It's just a different way to show it. If you look at GGG on here, here's how this works. This chart, this is the first letter. This is the second letter. This is the third letter. So if I have GGG, I would say, all right, my first letter is G. That means I'm somewhere in this bottom 4 rows here. The first letter being G. The second letter being G means I'm somewhere in this column of this row, or of these 4 bottom rows. And the third letter being G means I'm in the bottom column, the bottom row of that column. So G, G, G gives me this bottom glycine right here. But notice, I could have spelled glycine many different ways. So again, some mutations, some changes in the genetic code will really have no difference. I could have spelled glycine G, G, U, still get glycine. G, G, C, still get glycine, and so on. So again, sometimes changes don't make any difference. Sometimes the differences are very, very significant. All right, let's go down to here, shall we? Here is our point of interest for today, mutations. Mutations. What are mutations? They're simply changes in the genetic code. There are a couple different kinds. Actually, there's like big scale mutations and there's small scale mutations. Think about if you're writing a paper and you have one little letter that gets changed in your paper, right? One little letter of one little sentence. That would be like a small scale mutation, what we would call a point mutation. Some mutations are much, much larger, and they would involve entire maybe chapters or paragraphs of your paper that you're making. We'll get into those in a little bit. But today we're going to focus, or right now we're going to focus on point mutations. Point meaning a single base pair or a single base in the genetic code is changed. There's a mistake. And again, this can occur when DNA is copying itself to make a new cell. This can also occur when DNA is transcribing or even translating into RNA. So one nucleotide, very important vocabulary here, point mutation. One nucleotide in a codon is substituted for another. That's one type of point mutation. And these generally result in only one amino acid changing in a protein. If any amino acid, sometimes it won't even change one amino acid. When a point mutation results in a deletion or an insertion, this would not be a substitution now. This would be a change, or I'm sorry, this would be an addition or a omission of a nucleotide. That's going to cause what we call a frame shift, and I'll show you that in a second. But keep the big picture in mind. Remember, DNA ultimately leads to our traits. So if I have a change in my DNA code, I could get a change in my protein, which could give me a change in my chemical reaction, which could give me a change in my trait. And again, this can lead to variety and diversity. Okay, let's have a look at something a little bit more technical now. Follow me close here, please. This is kind of complicated, but not too bad. Let's say that this red strand right here was the original left side of my DNA. This is what my DNA was coding. Okay, it's like Miss Hager. And this bottom row right here is my messenger RNA. And my messenger RNA, as you know, I can use, and my cells could use, to translate the message into a specific amino acid, right? So let's say that there's a mistake in my original DNA. All right, so right now, this would be normal. This would be the original unmutated process here from going from DNA to messenger RNA. So if this was my original DNA, this red across the top, and this purplish blue was my RNA across the bottom, and you guys remember the base pairing rules. T will go with A will go with U. Remember, DNA doesn't have any uracil, and RNA doesn't have any thymine. So anytime you want to put a T here, you put a U. Anytime you want to put a U here, you would put a T. In other words, DNA has thymine instead of uracil. RNA has uracil instead of thymine. So if this was my original DNA across the top, that would make this my messenger RNA across the bottom here, the bluish purple. That code would give me these particular amino acids. And if you don't believe me, you could go back and check me on the wheel. And if you read those codes in 3 letters at a time, you would find, whether you want to use this chart or you want to use the wheel, you would find that the amino acids that would come from that code would be these: methionine, threonine, histidine, aspartic acid. I believe that isn't glycine. Excuse me. However, let's look at this. If I had a mutation where this letter got changed, this letter got substituted. Notice there used to be a T there, but now there was an accidental misspelling and it gave me an A instead of a T. Notice the rest of the strand is fine, just like it was up here. Well, if I get a mutation or a mistake in my DNA, that's going to cause whatever sticks to it, messenger RNA, to also have a mistake. So if you look here, this T changed to an A. It was a mistake, which means this A is going to change into a U. Well, if you look at how that codon, or how those codons now translate into amino acids from using the codon wheel, you will see that this second amino acid used to be threonine right here. Now it's serine. And again, you could go back and check that on the wheel. If you don't believe me, just go back and look those things up. You can use the chart or you can use the wheel. Okay. That mistake, let's look what it resulted in. Please remember, again, DNA and RNA are read in groups of letters of 3 at a time. So my first codon, my first DNA code was TAC, meaning my first codon was this AUG. Remember, think of putting like little lines every 3 letters, little frames of reference every 3 letters, okay? Because each 3-letter code gives me a specific amino acid, okay? So in this case, notice the first 3 letters weren't changed in the DNA, which means they weren't changed in the RNA. However, the 4th letter was changed. It was substituted. 1 letter that came in to substitute for another letter. Notice there's not a missing letter. There's not an inserted letter. There's just a switch of letters. One substituted for another, just like a substitute teacher would come in to teach for your class. You would still have a teacher there, but it would be a different teacher, a substitute, okay? So notice now that the second 3 letters, remember, genetics is always about 3 letters, what we call codons. That second word or codon is now AGG, whereas it used to be TG. G. So that means the RNA, instead of being ACC like it used to be, is now going to be UCC. And the translation of that is not going to be threonine like it used to be if you look at the codon wheel. Now it's going to be serine. So as you can see, that was the only change in the mutation or in the strand of messenger RNA, meaning the only change in my protein was that second amino acid. It used to be threonine in the original. With the mistake, it's now serine. However, everything downstream is still the same. Histidine, aspartic acid, glycine, histidine, aspartic acid, glycine. Okay? Now, what if this occurs though? Wonder if I have an insertion or a deletion, meaning not a substitution of a letter, but where there's a letter actually missing or a new letter added into all the other letters that are already there. Well, that's going to cause some very interesting problems or some interesting changes. And let's take a look at what that could cause.
Ever since I was a kid, I have been curious about mutations. I think certain movies may have had an impact. I used to imagine mutations with amazing abilities, but there was also a lot I didn't understand about mutations. Let me explain. Many people understand that a mutation is a change of genetic material, more specifically, a change within a nucleic acid. RNA and DNA are both types of nucleic acids. Therefore, anything with RNA or DNA can have a mutation. That means animals, and that definitely includes humans, also plants, fungi, protists, bacteria, archaea, they can all have mutations. Additionally, so can viruses. Many mutations can be neutral in effect. You can see this codon codes for the amino acid leucine. But if it experiences this particular silent mutation, even though a base has been altered, it still codes for leucine.
Okay, so keep in mind here, some mutations can cause really no effect because there are different ways to spell the same word, right? Like quick and fast, or John and Johnny and Jonathan. So in this case, this is called a silent mutation. The change in the DNA and or the RNA didn't cause a change in the protein. No effect.
It did not change the amino acid. Mutations can also be harmful or helpful, but it's important to understand that mutations are random. The organism can't will itself to get a certain mutation. You can learn more about this in our natural selection video, where we talk about how a bacterium may already possess a helpful mutation that allows it to survive an antibiotic. But the bacterium didn't will itself to mutate to develop this certain mutation. Okay, so again, mutations are random. But there can be factors that make mutations more likely to occur.
I want to stop right there and just point out, especially with this coronavirus stuff, mutations change the genetic makeup of organisms. And that can make certain diseases harder to treat, because as we get medicines that can treat these organisms, they mutate and they change, and then our medicines no longer work to kill these germs. So it's an important thing to realize that mutation causes diversity and causes variety, and sometimes that's a good thing, sometimes that's a bad thing, especially when we're talking about germs like viruses.
External factors like certain types of chemicals or excessive radiation, or internal factors like an event that causes a problem during DNA replication in interphase. During interphase, cells can replicate DNA before they divide. So let's discuss some different types of mutations. We'll start with gene mutations. DNA makes up genes, and genes can code for proteins that influence different traits. So when a mutation in DNA happens, which specifically means a change in one or more DNA bases, then different proteins can be produced, which can affect an organism's traits. In this example, we have fruit fly DNA. Mutations could include substitution, which means the wrong base is matched. Insertion, which means an extra base or bases are added in. There is also deletion, which means a base is removed.
Okay, keep this in mind. This is really important screenshot here. Substitution is simply a swap out of one base for another. Insertion is a brand new added in between other existing bases. And deletion is a base is missing from the entire strand or chain or code. These are very different. These are all known as point mutations because they only involve one genetic base, like an A or T or C or G or a U, right? They are small scale mutations, like in your paper, if you're writing a paper for English, they're just maybe one letter in your paper that has been has had a mistake occur. Point mutations: substitution, insertion, and deletion. Point mutations.
Insertions and deletions have the potential to be especially dangerous. Why? Remember in protein synthesis how we talked about how bases are read in threes? Well, if you add a base or remove a base, suddenly the number of bases total has changed. And if you read the bases in threes, depending on where it happened, everything that is read afterwards could be affected. We call this a frameshift mutation. If you look at this frameshift example here, where a single base has been inserted, you can see how the following codons, which have 3 bases each, are now all.
Notice here, the original DNA strand, the original messenger RNA strand that would have stuck or base paired with that process called transcription. Remember, this is like Miss Hager. This is like the pink slip, okay? If you change one letter, it might not be a big deal. However, if you insert one letter, that will make this entire code shift over by one letter. And now remember, we're reading in groups of 3 nucleotides at a time. So if a new letter is inserted, that shifts the entire reading frame of the messenger RNA and the DNA, and that's going to give me different amino acids than the originals were supposed to be, okay? Don't worry, I'm going to explain this a little bit better later. Here's that chart instead of the wheel, same information though.
Affected as the reading frame has been shifted. This example shows how it can lead to many amino acid changes. There are also chromosomal mutations. Remember that chromosomes are made up of DNA and protein, highly organized, and they have lots of genes on them. The human chromosome number is 46, with 23 from an egg cell and 23 from a sperm cell.
Okay, so now we're going to talk about entire chromosomes being mutated or changed. Remember, a point mutation was like a single letter in your paper that you were writing for English class, a single letter being changed. If you think about chromosomal mutations, these are entire chapters or maybe paragraphs or pages, large chunks of information that are either inserted or deleted or substituted or relocated. It can get very complicated, but just keep in mind a chromosomal mutation is very large scale. Think of chapters.
This type of fruit fly here, however, has 8 chromosomes, so 4 came from an egg cell and 4 came from a sperm cell. Some examples of chromosomal mutations include duplication, where extra copies of genes are generated, deletion, where some of the genetic material breaks off, inversion, when a broken chromosome segment gets inversed, which means reversed, and put back on the chromosome, or translocation, when a fragment from one chromosome breaks off and attaches to another chromosome.
Okay, so look at this. Again, this is big scale stuff. This is like entire chapters. These are chromosomal mutations. Duplications, where maybe some of the pages in your chapter actually repeat themselves now. And if you think about the DNA code and the chromosomes as being instruction manuals, that could cause some major changes if you have repeated pages in a manual where you're trying to build a product. Like let's say you're trying to build a bicycle or something, and all of a sudden now you have the same pages in the manual occurring 2 or 3 times. That could really confuse you, right? And the same with cells. Deletion, again, wonder if an entire page is just gone from your instruction manual. That could really mess with you as far as when you're trying to build your bike. Inversion is just kind of a reversal. In other words, wonder if maybe chapter 5 and 6 now appears in your book. Chapter 6 comes first and then chapter 5. Again, these are large scale chromosomal mutations, and they can cause a lot of change very quickly. And sometimes they can be actually good once in a while. Sometimes it can be very bad. Finally, translocation. Wonder if some of the pages from your chapter 7 are now in your chapter 10 Well, that would really, again, confuse you if you're trying to follow that instruction manual and trying to build something.
Chromosome. There's more mutations than what we covered, of course, but the idea is that there are many different kinds of changes that can happen. If a mutation is going to happen, we already mentioned that there are especially vulnerable times, such as during DNA replication, but also there are other times too, like during meiosis. In fruit flies and other animals, meiosis makes sperm and egg cells that can have the number of chromosomes as the organism. However, sometimes those chromosomes don't separate completely. We call this nondisjunction. This can result in an egg or sperm cell that has too many or too few chromosomes.
We talked about this in my class. This is known as like a trisomy or a monosomy. Some of your sperm or egg cells can end up having, instead of exactly half of the chromosomes that they should have, remember sperm and egg cells should only be 50% of your DNA. Sometimes if the split is not equal when you're making sperm or egg cells, some of your sperm or egg cells could end up with an extra chromosome, and some of your sperm or egg cells could end up with a missing chromosome. Well, remember, these cells are what's going to lead to your baby ultimately. And if your sperm cell or egg cell is missing a chromosome, that means every cell in your baby is going to be missing a chromosome. If your sperm or egg cell has an extra chromosome, that means all the cells in your baby are going to have an extra chromosome. And we talked about these different conditions. Again, this would be called a monosomy, not enough chromosomes or missing a chromosome. This would be called a trisomy, meaning you have an extra copy of a chromosome. Probably the most famous trisomy is Down syndrome, which is known as trisomy 21. That is a chromosomal duplication. You have too many chromosome number 21s. You have a third copy instead of just having two copies like you should.
It's possible for a mutation to be passed down to offspring. Consider a protist with a mutation. Many protists reproduce asexually, and when they divide, the daughter cell can inherit the same mutation. A fruit fly, which reproduces sexually, can pass a mutation to its offspring if that mutation is found in the genetic material of the sperm or egg cell. You may wonder why we've been mentioning fruit flies so much. Well, they're awesome, but also it turns out that fruit fly mutations and how they are inherited are frequently studied. We have some links in the video description for more info. These studied mutations can also occur in humans, and we'll give a gene mutation example of a substitution in the case of sickle cell anemia. First, a little background. Hemoglobin is a protein in your red blood cells that helps you carry oxygen. But in the disorder sickle cell anemia, the gene that codes for hemoglobin is mutated. If you inherit two copies of this gene, one from each parent, you can have this disorder. This disorder can make it difficult for your red blood cells to carry oxygen because the shape of the red blood cell is affected from this mutated hemoglobin protein.
Notice this mutation here. This was the normal DNA. This was the normal messenger RNA. These were the normal amino acids that produced the normal protein that gave you the normal red blood cell shape. This red blood cell shape is important, by the way, because it carries oxygen right there in that little donut hole of your red blood cells. Look at this, very interesting. In sickle cell anemia, there is only one little misspelling here. There's an A where there used to be a T. One mutation. That's called a point mutation. That's back to that small scale, one letter only being affected. Don't be confused here. Even though it's only one letter, it can still have the potential to cause a very large change in your protein that's being made. So remember, if I change my DNA, it's going to change my messenger RNA, which could then change the amino acids that are put in that correct order, and that could change the trait. So if you follow this mutation here, that T mutated to an A. By the way, ask yourself, is that a substitution? Is that an insertion? Or is that a deletion? Well, I'm sure you'll realize that's a substitution. One letter got changed to another letter. Still, the correct reading frame is taking place. We're still reading 3 letters at a time in the right frames or the right order. However, just this one little mutation here causes a big change. So it used to be the second codon here, the second word in DNA was CTC. Now it changed to CAC. Well, remember, that's going to cause a change in the RNA, the messenger RNA. Instead of being GAG, that would have paired with that DNA up there, now it's going to be GUG. Well, you might not think that's a very big deal, but if you go back to the codon wheel and you see what that translates into for the amino acid sequence, instead of glutamic acid, which GAG would give me, again on that codon wheel, if you read that codon wheel for GUG, it's not glutamic acid anymore. Now it's valine. So now I have a different amino acid in that number 2 spot of that protein. That's like Miss Hager having a mistake, writing the wrong pink slip, and going to get the wrong child out of class to bring down to the office. That's a very
This can
important
lead to
change.
anemia and other problems. Well, unfortunately, there is not a cure for this disorder yet. The good news is that treatment for this disorder has greatly improved. Another thing to mention, if an individual only inherits one copy of the mutated gene from one parent, they are a carrier, but they don't officially have the disease. Usually they do not have symptoms, but those that are carriers appear to have a protective factor against malaria. Malaria is a disease caused by a protist that can be transmitted by mosquitoes. These individuals can still get malaria, but their symptoms are often less severe. We should mention that studying mutations and genetic disorders is a large and important field right now. Genetic counselors work to help families that may be affected by genetic disorders. If you have an interest in learning more,
Okay, we're going to stop right there. Back to our PowerPoint. Okay, so you saw some of these different mutations. All right, I'm going to talk to you later about a frameshift mutation a bit more. This results in a reading frame change. This shifts the entire sequence of DNA or RNA being read by maybe one letter or two letters, and I'll explain that to you in a second. So frameshift mutations are mutations that result in a protein not being able to perform their duties. However, you got to remember, sometimes if the protein is changed, it might actually do a better job than it did originally. So keep that in mind. And finally, chromosomal mutations. Again, entire sections of chromosomes containing many genes. Frameshift mutations, these different point mutations that we talked about up here, these are smaller scale. They could still cause a large change, but these chromosomal mutations are large scale. Again, think of them as chapters, okay? Let me show you an example of that. Chromosomal mutations. This is like an entire chromosome in any organism, right? A deletion in a chromosomal mutation would be like leaving out part of an entire chapter of information. So look at my original chromosome, should have had A, B, C, D, E, F, G, H, I. Maybe those were the pages in that chapter, right? The genes on that chromosome. And now, after deleting part of it by accident, my new chromosome has has pages A, B, C, H, and I. So these D, E, F, and G pages are missing. Major changes. Let's look at chromosomal duplication. Again, these are all large scale stuff. Think of them as chapters again. Duplication for a chromosomal mutation. My original chromosome should have had pages A, B, C, D, E, F, G, H, and I. And now, when the chromosomes replicated, perhaps, I got an extra C and D in there. So now my pages are A, B, C, D, C, D again. So I have those pages occurring again in my instruction manual, E, F, G, H, I. You can see how this would be major change, okay? Inversion on a chromosome. When chromosomes are being copied, sometimes the pages get switched backwards. So instead of having pages A, B, C, D, E, F, G, H, I in the right order, I have A, B, C, F, E, D, G, H, I, because these pages, D, E, and F, got flipped around backwards. Again, imagine the confusion that would cause you when trying to build your bicycle or whatever that instruction manual you had was. Remember, DNA is the instruction manual for your cells. not only on how to build the cell, but how to maintain the cell throughout its life. So these can be major changes. However, again, sometimes you get lucky and these changes actually end up being better. I'm sure you guys have all made a mistake before that ended up being a good thing. That happens in genetics as well. Okay, translocation. Notice now we're talking about different chapters. Imagine your chapter 6 or your chapter 1 now contains pages from chapter 2. Well, that would be goofy, right? Or your chapter 2 contains pages from chapter 1. Again, that would cause confusion. All right, let's have a look at another little quick video here.
The differences in our genes come about through a natural process called mutation. Mutation is extremely common. As a cell copies its DNA before dividing, it makes occasional typos. Most commonly, a single base is substituted for another. Sometimes a base is deleted, or an extra base is added. The cell is able to repair most of these typos, but each round of cell division introduces a few changes that are not repaired. When DNA changes occur in cells that will give rise to eggs or sperm, they can be passed down to offspring. Each one of us has about 60 new variations that were not present in our parents. People commonly use the terms mutant and mutation to describe something undesirable or broken. But mutation is not always bad. Most DNA changes fall in the large areas of the genome that sit between genes, and they usually have no effect. When variations occur in areas of the genome that code for proteins, they often have consequences. These changes can affect the protein product itself, or they can affect when, where, or how much protein is made. Most of the time, mutation generates variations that are neither good nor bad. Just different.
Okay, interesting. I hope you can see that not all mutations are bad. Not all mutations even have a real effect. Okay, I'm going to point this out to you finally. You guys know how I love my analogies. So if you hear anything today, hear this. Mutations, genetic mistakes. Remember, mistakes aren't always bad. Couple different kinds of mutations or a couple of different categories of mutations. We just talked about this. This is kind of summing it all up for you. Point mutations. Point mutations are simply mutations that involve only one letter, one nucleotide base. So an A, a T, C, G, or a U, okay? But remember, DNA/RNA is always read in bases of 3 at a time, so groups of 3. So if a base is like a letter, a group of 3 letters is like a word in the genetic code that we call a codon. And that's what that whole codon wheel was based on. Remember, codons are really just codes for amino acids. They're codes on what amino acid to put where to build the protein. If you want to go back to the Miss Hager analogy, codons are simply like what student should go in the right order of that student group, the top 10 for that picture that we took for the yearbook. And if you don't know what I'm talking about, that means you didn't listen to the other PowerPoint videos. You need to go back and do that, okay? So types of point mutations. One type is a substitution, all right? One letter is substituted for another. Now follow my reasoning here, okay? If you look at substitution mutations, it's simply one letter got swapped out for another. So remember, again, the genetic code is read in letters of 3 that we call codons. I made a silly example here for codons, but you get the idea. Instead of, A, T, C's, and G's, I just wrote different 3-letter words in the English language, right? So let's say you had a sentence, the red cat ate the fat dog, pretending that's a genetic code for something. And let's say I swapped out just the one letter here that I have in red, the R. The T got substituted for an R. This is a point mutation called a substitution. Only one letter was involved. Only one nucleotide base was involved. So now my new sentence, instead of the red cat ate the fat dog, my sentence now becomes the red cat ate the far dog. Well, that changes the sentence, but it's not a huge change. So you still kind of get the gist of the sentence or the meaning of the sentence. So a small difference, maybe no effect on the actual cell or the body or whatever. But then look at this. We could have that same kind of point mutation, that substitution, one letter for another letter, and it could have a much bigger difference. For instance, go back to the original sentence. The red cat ate the fat dog. Well, let's say that my mutation was the C got swapped for a B here in red. Now, instead of the red cat ate the fat dog, it becomes the red bat ate the fat dog. Well, that's a big difference. If you're trying to tell a story or something, a bat is very different than a cat. So even though it's only one letter, it could cause a big difference in the meaning of that sentence. So even point mutations that are substitutions can cause, kind of large changes. Again, this might not be a bad thing. It could be a good thing. It could have not much of a difference at all. But let's look at some other point mutations. Again, point mutations are just one letter difference, but there are different ways to change letters. Up here, we just swapped one for the other, like a C for a B. But look what happens if we add a letter, an insertion mutation. It's still a point mutation. It's still only one letter, but my gosh, could it cause a big change? This is insertion is an extra base being added in or inserted into the genetic code. This causes, make sure you know this word, a frame shift mutation. This is a frame shift mutation. Remember, the words are always read in groups of 3 letters. So if I add a letter, it's going to shift the entire reading frame down towards the right, just like when you read a book. So watch this. The red cat ate the fat dog. That was my original sentence, right? But let's say I insert a Y to this sentence. I'm not substituting a Y, I'm inserting it. It's an extra letter. Well, look what happens with the meaning of this sentence now. Instead of the red cat ate the fat dog, it becomes the red cat yet, if, ifa, to the g, which is silly, but you understand the idea, right? You can still see the original words in there, right? You can still see ate the fat dog, but when your genetic code is being read, it's read in groups of 3 letters. So everything has shifted over towards the right. This is a frameshift mutation. You can imagine that this would change all the amino acids that your protein is trying to build. This would be a massive change, perhaps even destroying the protein or not allowing the protein to be built. Let's look at another frameshift mutation. Remember, these are still point mutations. These are still point mutations that are only involving one letter, but there's different ways to do this. So the substitution didn't cause much change at all. The insertion caused a frame shift. Everything got shifted to the right. Let's look at a deletion. A deletion, again, still a point mutation, only one letter involved. However, it's going to cause another frame shift. So I'm not substituting a letter. I am, in this case, removing or deleting a letter. So the red cat ate the fat dog. That was our original sentence. And now it becomes, if I, let's say, get rid of this C, the C is gone. It's deleted. Now the frame has shifted. So my new sentence becomes the red ata, because that C is gone now. So now I start reading the next word at the letter A. So the red ata, tet, hef, ata de ag. It makes really no sense anywhere downstream or to the right of that mutation. So again, these 2 frameshift mutations, whether it be insertion or deletion, these both cause frame shifts where the entire reading frame is shifted over and anything downstream, anything to the right, is affected, okay? So since insertions and deletions both cause a frame shift, they usually cause more dramatic change or difference in the protein and therefore in the trait being made. Again, though, sometimes that actually turns out to be a pretty good thing. Chromosomal mutations, remember, these are kind of the opposite of point mutations. not really the opposite, but they're just larger scale. Chromosomal mutations are like entire chapters of information being deleted, inserted, relocated, shuffled around, put backwards, you name it. A lot of different things can go on. However, I'm sure you can see this leads to a lot of variety. This is why things in life are different. Remember, all life on Earth started with one cell, one species, and now we have millions of species. And the reason for that is partially because of mutations. These cause changes. So we're guaranteed to change over time. And that's a good thing because what kills one won't kill all. And some species will be able to adapt and not go extinct because of this. We can change over time through evolution. And that's partially thanks to mutations. So don't think of the word mutation as a bad thing all the time. So to summarize, point mutations are changes in only one nucleotide base, one letter, A, T, C, G, or U. They may cause no change, they may cause very little change, or extreme change in the protein that's being built, which, by the way, would cause an extreme change in the trait, because the proteins lead to the traits. It really all depends on what the mutation is and where on the gene that it occurs. Going back up to here, you can imagine if these mutations, let's say these insertion and deletion mutations here, the earlier in the sentence that they occur, the more they screw up the sentence. The later in the sentence they occur, let's say it occurred right here at the word dog, well, everything to the left of that would have been okay, it would have been normal. So not only does the mutation or the severity of the mutation depend on what it is, it also depends on where it is in the genetic code or the sentence. All right. And finally, point mutations that cause a frame shift can drastically or completely change the protein being made. It may cause a situation in which the protein is too short, too long, or never even made. Let's go back up to this. Wonder if you didn't have your capital letter here to start your sentence. Your genetic code needs a capital letter as well. Just like we have punctuation in our language, the genetic code has punctuation. So if you have a punctuation issue for a start codon, that could really screw things up because you would never start to read that genetic code and you would never start to make that protein. Or let's say you had a punctuation issue and you should have a period right here and you don't get the period in the sentence. That would mean the next sentence would run on into the following sentence. And again, that would really screw things up very, very severely or change things very severely. So big picture here, it depends where in the sentence or where in the genetic code the mutation occurs. Okay. So mutations are sometimes bad, sometimes have no noticeable effect, and sometimes are actually good. Mutations lead to variety in all organisms, sometimes creating new traits. And believe it or not, over time, these new traits can add up to creating new species over time. All right, I hope that helps, everybody. I'm going to get you back to my mugshot here. I hope that helps you guys. I hope you have a great day. Tell your mothers happy Mother's Day, Sunday. And please make sure you do a good summary of this video depending on what your instructor's instructions were. We miss you guys. We hope we'll see you in the fall. We're almost done. Keep doing your work. Keep making up missed assignments. Remember, it's more important to go in order than it is so much to get them in on time. Okay? Have a great day, everybody.